In lactase non-persistence, also known as lactose intolerance, you have the disaccharide lactose, which is a disaccharide, two monosaccharides combined. Those two monosaccharides are a beta 1,4 configuration of glucose and galactose. And they are not broken down by the lactase enzyme because, well, you don't have it. So that's not broken. You are not able to transport glucose because there is none. So lactose, which is a disaccharide, there is no transport membrane for it. There is no transport membrane transport protein for it. So lactose stays in the gut. And guess what it is now? Salt. And what's water going to do? Water follows salt. Water follows salt into the gut tube from your cells. And then water is going to follow the gut tube out the only exit it knows how to. I'm going to leave that to your imagination. And this is the internet. The internet has a big imagination. Okay. So, here's what happens to your cells. In a cell that is in a water potential that is equilibrium with the cell's water potential, the cells will stay their correct shape, their normal shape. These are blood cells, by the way. You can tell by their circular biconcave disc appearance, the fact they have no nucleus, that they are red due to the function of hemoglobin carrying iron, which carries oxygen. And when you place them in distilled water, which has a low salt concentration, by low it should be none, because that's the point of distilling it. But anyway, when you place them in distilled water, the cells will swell eventually to the point of bursting. This is called plasmolysis. Plasmolysis is damage to the plasma membrane due to osmotic forces. So water enters that cell following the salt, because your red blood cells, they're quite salty compared to distilled water, and water follows the salt. Now, when you place the water into a concentrated salt solution, water follows salt, but the salt is now outside the cells, whereas the water now inside the cells. So the waters are going to come from the inside of the cells, follow the salt to the outside, and the cells are going to shrivel and shrink. They're going to become crenated. I'm going to spell that for you. We're going to spell that C-R-E-N-A-T-E-D. Crenated. It's probably not going to come up, but just in case, you might as well spell it correctly, right? Okay. Well, you can pause. Remember, K button. Now, here is a series of images. We have normal appearing red blood cells with their circular biconcave appearance. As you increase the water potential, the RBCs will fill and they'll lose that biconcave appearance, becoming spherical in shape. Eventually, they will either lyse or fragment, and you'll get these RBC ghosts. These RBC ghosts are the leftover plasma membrane sacs that you basically don't have anything left in them anymore. And as you decrease the water potential, increase the solute concentration, the salt concentration, they'll become more and more crenated. And the water will be leaving the cell to follow that salt. To follow that salt all the way to LA to pursue its movie career. Alright? Leaving behind everything. Now, in the plant cell, we have a different situation. Not that different, mind you, because when you put a plant cell in distilled water, the cell will still swell. And when you put it in salt solution, it's still going to shrivel and shrink and become crenated. However, the difference here is that plant cells, because of the function of their very stiff, fibrous, beta glucose cellulose cell wall, they do not burst. Plant cells do not burst when you put them in distilled water. Instead, they just swell and become turgid. But here, This is plasmolysis for a plant cell, when the plasma membrane is damaged by being peeled away from the cell wall. So plasmolysis in a plant cell is when you put them in a high solute concentration solution and the plasma membrane is peeled off. Now, on a lab exam, you would draw a plasmolysed plant cell. You would draw a plasmolysed plant cell like that. And if they ask you to label something, then you would either label, if they ask you to label the cell wall, then you're going to label that cell wall. If they ask you to label the plasma membrane, you're going to label that. Plasma membrane. So that's what it's going to look like on a light microscope image. And that's how you draw it. You won't be able to see individual organelles. Your microscopes are not that good. Because, quite frankly, no light microscope is. That the size limit of visual distinction on a light microscope is 200 nanometers in size. And also you need to stain something in order to see the nucleus at all. So if you stained it with iodine, you might be able to see the nucleus. But that's not always going to be the case. Oftentimes you'll only be staining it with salt water and you'll just see the plasmolysed cell. Now, let's talk about how molecules cross the plasma membrane through active transport. So active transport. Remember that slide before about the sodium and the potassium and the calcium? They must be maintained at very steep concentration gradients inside and outside the cell. Cells really need to maintain very high or low levels of some molecules in order to survive. Active transport proteins are used to pump these molecules in or out of the cell. You require energy in order to pump. That energy is provided by the hydrolysis of ATP, typically. Typically, we are hydrolyzing ATP in order to obtain this energy. So, we start this way. Let's say that we want to keep things this way. Well, that means that we're going to need to expend energy by burning ATP or breaking down ATP in order to push these molecules out. And to take these molecules in, we're going to need to expend ATP. We're going to need to pay ATP. So, in order to maintain and get this circumstance, we need to burn ATP. We need the sweet, sweet energy in those anhydride bonds, those phosphate oxygen, those phosphorus oxygen phosphorus bonds. That sweet, sweet energy that we obtain through hydrolysis. Now, there are a couple of different types of transport membrane transport proteins. There's your typical carrier molecule, which you take your energy particle, which the ATP can service, the hydrolysis of ATP can serve as your energy source. And you take your molecule that you need to excrete and you supply the energy and you excrete the molecule, or you take in the molecule. This could be the inside or the outside. This could be the outside or the inside of the cell, and vice versa. It doesn't matter which side is which. If you need to pump something into the cell or out of the cell, it just depends on what protein is doing what. Now, there's also coupled transporters. And these can also be passive. They don't have to be active, but for our purposes right now, we can talk about these as active transporters. We have symporters, where our two substrates are coming in from the same side of the membrane. If they're both coming in from the outside, for example, hydrogen and calcium. If hydrogen and calcium are both coming in from the outside of the cell membrane, or from the outside of the cell membrane, that's a symporter. If an antiporter is in play, for example, sodium and potassium, where one is being kicked out and the other is being brought in, that's an antiporter. Anti, opposed, sym, with. So a symporter takes two different molecules and brings them into or out of the cell together, and an antiporter exchanges two molecules for each other. Now, it doesn't have to be 1 for 1. It could be 2 for 1. It could be 3 for 2. It could be 5 for 10. It doesn't matter what the numbers are, as long as you have an exchange that is occurred. So, this is your typical carrier protein at work, just moving stuff from one side to another, and this would be an antiporter. This would be an antiporter at work. Molecules moving in, molecules moving out. Now, this is a summary table that we've already discussed, and now let's talk about bulk transport for a moment. Endocytosis is the process of taking substances into the cell. Taking substances into the cell. There are two subsets of endocytosis. There is phagocytosis and there is pinocytosis. Now, I pronounce it phagocytosis. I have heard it pronounced phagocytosis or phagocytosis. I am not picky. I am accepting of all pronunciations, as long as you spell it correctly. It's not spelled with an F. It's spelled Phagocytosi. Alright, so phagocytosis is a type of endocytosis where an entire cell or a cell fragment is engulfed. Some sort of enclosed vesicle or body is engulfed. Pinocytosis is cell drinking. So phagocytosis is cell eating. Pinocytosis is cell drinking. The external fluid of the cell is engulfed, and certain particles are taken in along with that cellular fluid. Now, receptor-mediated endocytosis occurs when the material to be transported is binding to certain specific molecules on the membrane. Now, pinocytosis is really important for this. So we have on our membrane certain receptors for stuff. And they're going to hold on to molecules that we want, and then eventually, when we want to take them in, we're going to make an endocytotic vesicle that also will have those receptors on it. So eventually, we'll get we will get our engulfed vesicle with our stuff. That's pinocytosis at work for receptor-mediated endocytosis. Now, we could also have receptor-mediated endocytosis helping us with phagocytosis. That's not a problem. It works too. But that's what's going on here. But in general, endocytosis works in this way. A food particle is taken in by endocytosis. We have a food particle. Whether or not this food particle is just a bunch of fluid, or it's particular things that are attached to the receptors on the cell surface membrane, or if it's an entire bacterial cell. It does not matter what that food particle is. An invagination in the cell surface opens up. So we have this particle being engulfed. That will pinch off, forming an endocytotic vesicle. This endocytotic vesicle will fuse with a lysosome. The lysosome being an organelle that we learned about earlier on in this unit, and that will form a secondary lysosome. Now, the lysosome contains lytic enzymes that are designed to cut up and take apart and tear away all the stuff inside that phagocytotic, that endocytotic vesicle. That will digest whatever's in there. Now we have this secondary lysosome filled with digested food particles. Now, through a process of very careful picking and active transport and what have you, we're going to actively transport or diffuse or osmose out whatever we want from this bag of digested sack of stuff. Basically broken up chips, I guess. Yeah. We're going to diffuse out what we need from this. And that bag is now waste. This vesicle is now a bunch of waste. That will now be put back to the membrane to be excreted by exocytosis. Endocytosis, if you remember your Greek back from Unit 1, is taking in. Endo, within, cytosis, cyto, the cell, osis, process of. So endocytosis is the process of taking things into the cell. Exocytosis will be exo, exit, or leaving, or outside. And cytosis, the process of making things go outside the cell. So this is dumping the waste. So exocytosis is going to be fusion of this vesicle to the plasma membrane, and then dispersing of the particles of waste into the extracellular environment. So, the phagocytosis is just a specialized form of endocytosis, where the cell can, in fact, engulf something nearby that is large and probably doesn't want to be engulfed, let's say. So this would be vesicle-mediated transport. Vesicles and vacuoles that fuse with the cell membrane can be utilized to release transport chemicals on the cell to allow them to enter a cell. Exocytosis is the term that is applied when the cell transport is out of the cell. So a vesicle here fuses with the membrane and is dumped. So fusion, excretion. Alright, this is just another option. Neurotransmitters being released from a synaptic vesicle work in this way. Now, this slide we don't worry about. Pause the video.